Hello everyone, and welcome to episode 99. This is going to be a really cool show because we're going to be talking about some really strange creatures. But let me go over a few things first. In the previous episode, we explored the evolution, anatomy, and ecology of the sponges of the phylum Porifera. Today's show will continue with our series on these early metazoans by exploring another extremely basal and particularly cryptic group of animals, the Ctenophora, or the comb jellies. Okay, right at the outset, let me just say that these guys are super strange. They kind of look like semi-translucent, neon-colored trash bags floating in the water. They're much more morphologically and behaviorally diverse than the sponges, but they're seemingly simpler than the cnidarians, the jellyfish. They do not have fins, but instead they swim through the collective force of thousands of cilia arranged in rows along the outer surface of their body, making them the largest animals to transport themselves with such small appendages. Those rows of cilia look like combs, giving the creatures their common name. So without rambling too much, let's just get right into it. We'll start with the evolutionary history of the Ctenophora, followed by a very brief overview of their taxonomic diversity. Then we'll take a look at their anatomy and physiology. At the smaller scale, we'll look at their cellular dynamics and their biochemistry, and at the larger scale, we'll look at their diet, their behavior, their reproductive processes, and their ecological relationships with other organisms in their environment. It'll be a deep dive on these strange, deep-sea animals. Let's begin about 580 million years ago, in the heart of the Ediacaran period leading up to the Cambrian. If you just listened to episode 98 on sponges, you should remember the main gist of the evolutionary history that built up the first metazoans. If you haven't listened to that episode, or episode 97 on early metazoan evolution, let me briefly summarize. In the time before the sponges, many different lineages of single-celled eukaryotic life had independently evolved multicellularity, like the ancestral plants and fungi. Related to the fungi, in a clade known as Opisthoconta, are single-celled choanoflagellates. These are little microbes with flagella that they use to move through the water and to create currents that suck up food particles for them to eat. We see really similar cells lining the inner tissue of the sponges, which use vast fields of choanocytes to create currents to move water through their bodies and vacuum up dissolved organic material. These flagellated cells are strikingly similar to their single-celled choanoflagellate cousins, creating many fascinating theories about the jump from single-celled to multicellular animal life. This seemingly straightforward relationship is complicated by genetic data, but in any case, the sponges remain the most primitive offshoot of the animal kingdom. And 580 million years ago, is when the first unambiguous fossils of sponges begin to appear in the fossil record. So we know they're at least that old, and they may belong to a lineage that's tens or even hundreds of millions of years older. The ctenophores are a different story entirely. Unlike the sponges, with their mineralized exoskeletons, the ctenophores are soft-bodied and jelly-like, so they're much more likely to be destroyed or to dissolve or be eaten by bacteria than they are to fossilize. Their fossils are comparatively rare, and we know relatively little about their evolutionary origins and about them as a clade. To illustrate this paucity of data about the ctenophores, consider this. I was born in the early 90s, and at that point in time, there had only been 2 ctenophore fossils discovered that were in good enough condition to actually be recognized and studied. It wasn't until I was 3 or 4 years old that more ctenophore fossils started to get discovered, like in the Stephen Formation in British Columbia, Canada. Some of the new ctenophore fossils were discovered in a nearby formation known as the Burgess Shale. These ctenophores were dated to around 505 million years old. Various lagerstatin deposits with ctenophore fossils have been found going back as far as 525 million years old. Now, this is still substantially younger than the oldest sponge fossils, but because the ctenophores rarely fossilize, and because their fossils are hard to find, it's highly likely that the ctenophores are much older than 525 million years. It's just that we haven't found the fossils. The evidence is just so rare and difficult to find. And our collection, our record, so to speak, is really sparse. There's just not much there. And this is really the tip of the iceberg of reasons why some scientists are hesitant to say for certain which lineage, the sponges or the ctenophores, is actually older than the other. In many cladograms and phylogenies of the early metazoans, you might notice that the Porifera and the Ctenophora are positioned together, but not in a way that shows which of them diverged first. They're often portrayed as being similarly distantly related to the rest of the animals, which means the origin of the sponges and the ctenophores and the precise nature of their relationship remains a mystery. The sponges are widely and traditionally considered the oldest animal lineage because, for example, they don't have mucin genes, while every other animal group, including the ctenophores, does. The ctenophores also have neurons that are very similar to jellyfish and sea anemones, but on the other hand, they lack Hox genes like the sponges. As we learned from the sponge larvae, they have a stage in their life cycle where they're roughly bilaterally symmetrical and they possess rudimentary nerves, but these are lost in the adult sponge. This would suggest some kind of regression or a loss of complexity, rather than a lineage of animals that have remained simple, unquote, for their entire hundreds of millions of years long existence. In the sponge episode, I noted the strong parallels between the choanocytes and the non-animal, single-celled choanoflagellates. Well, the ctenophores also make a strong claim to close relation with the choanoflagellates, as they appear to be cylindrical, somewhat like the sponge form, but turned inside out. The flagella are on the outside, but they're just smaller and more numerous. They're the cilia. But it can't be ignored that the ctenophores are also much more complex, with much more developed and varied tissues, and many more novel traits and life strategies. All of this just complicates the picture. And to make matters worse, as if it wasn't complicated enough, the ctenophores appear to have undergone an even more drastic loss of complexity of their own. For a long time, the question of ctenophore evolution remained a frustratingly enigmatic mystery. But as more fossil specimens were found, nebulous trends started to become apparent, and the backstory of modern ctenophores started to come into focus. The Cambrian fossils depict ctenophores that are somewhat more complex than they are today, with many more rows of cilia, and even veins or fins that extend from the main body, upon which the rows of cilia are mounted. They possessed larger bodies, with what appears to be internal organs, and they may have even developed specialized tissues, like organ systems, long nerves, a distinctive gut, and muscular sphincters that could close the mouth of the osculum. But all of these traits appear to have been lost in the modern ctenophores. One pathway down which the ctenophores did evolve was the achievement of free movement. Fossils in China, like the Stromatovarus and arguably the Dinomyscus, suggest that the ctenophores descended from sessile or immobile animals, or in other words, their ancestors were bound to the ground like the sponges. Where the sessile ancestor had cilia or flagella that it used to stir up water and propagate currents to extract food particles, the ctenophores appear to have totally repurposed these appendages by using them exclusively for movement. Instead of filter feeding, they've evolved an actual mouth, and their life cycle is defined by the slow-motion pursuit of tiny plankton and other smaller, weak creatures that can be easily captured. It seems like the ctenophores may have come second, descended from some sponge-like ancestor, or perhaps something like a frondomorph, or an ancient first draft of a crinoid. At some point, this population evolved to detach from the substrate, or perhaps their larvae evolved to retain their free-swimming lifestyle. In any case, they left the seafloor behind, and they began swimming freely in the open water column. These ancient ctenophores radiated and became quite abundant and diverse, but many of the older lineages, many of these early ancestral forms of the ctenophores, have all gone extinct. According to molecular clock data, all the modern ctenophores are descended from a crown group ancestor that existed around 300 to 400 million years ago. That's clearly a pretty rough approximation, but the estimates that I've seen suggest 350 million years ago plus or minus 80-something million years. That's a pretty big spread, so it's a rough guess. The lineages that diverged before this time, before this crown ancestor of the modern ctenophora, have all gone extinct. One such long-extinct group were the Scleroctenophora, which are known from fossils in the Maoshinshin shales in China's southwestern Yunnan province, and they date to around 510 to 520 million years ago. These are noteworthy because they possessed rudimentary internal skeletons, which is more data in support of that hypothesis that modern ctenophores have descended from much more complex ancestors. The ancient ctenophora had its golden age, its peak in abundance and biodiversity, around this time, hundreds of millions of years ago, in the late Ediacaran and early Cambrian. Today, they're a shadow of what they once were, but this is largely due to the fact that the oceans are full of all kinds of life today that just didn't exist back then. In the Ediacaran, the ctenophores were kind of like the latest cutting-edge technology. They were new evolutionary products with relatively few competitors and predators. In many cases, they were the predators. In contrast, in the Anthropocene, aka the modern day, the extant ctenophores numbered no more than 200 species, give or take. This low biodiversity is mirrored by surprisingly few genetic differences between species and clades. They're just not a diverse group, genetically or taxonomically speaking. But morphologically, how their bodies take form, what they look like, they can actually be very distinct. Let's briefly look at the ctenophore body plans to try to make these exotic creatures a bit more familiar. In all the ctenophores, no matter the body shape, the actual living visceral mass, the conglomeration of living cells working together, all form a 2-cell thick layer of epithelial tissue. To visualize how this tissue layer is arranged, imagine a transparent balloon with another smaller balloon inside of it. The skin of each balloon is like this epithelial tissue layer, lining the gut on the inside and the epidermis on the outside of the body. The air that's inside the big balloon is like the mesoglea, and the air in the smaller balloon is like that internal void cavity in the gut. It's the open cavity space inside the ctenophore's body. This balloon within a balloon metaphor is somewhat useful, but it's rough. It only goes so far. It has very limited descriptive value, as the ctenophore body is much more complex than that. For example, the gut cavity isn't its own sealed-off space. It's exposed to the environment through a large, open mouth orifice. The gut cavity also isn't smooth. It's not a smooth internal chamber or a capsule like the inside of a balloon. Multiple tunnels and channels extend outward from the gut, penetrating the mesoglea and creating watery corridors through which nutrients can be delivered to the most active parts of the epithelial layer, which is usually tunnels leading to the tentacles or an area rich with glands. 2 channels extend to the rear of the animal, to the aboral end, or the end opposite its mouth. These 2 channels perforate the epithelial cell layer and create anal pores or pseudo-anal pores. Despite their name, they actually release relatively little waste, as most digestive waste is expelled out through the mouth. The epidermis lines these internal channels as well, with ciliated cells in the mouth, in the pharynx, and the upper gut proximal areas that can all propagate water currents. Deeper in the channels, deeper inside of the gut tissue, closer to the outer epithelial tissues that they sustain with nutrients, you'll find various types of cells like storage cells that stock nutrients in their vacuoles, bioluminescent cells that create beautiful lighting effects on the ctenophore, and even gametogenic cells that produce eggs and sperm. I'll get into all of these topics, all of these cell types, in more detail later on in the episode. But for now, the oldest group of surviving ctenophora, including a plurality of known species, are the ctenopids. These have a generally spherical or egg-shaped body form, with 8 rows of cilia running along the length of the body, from the mouth to the aboral end. The ctenopids' larva possesses a pair of small tentacles that protrude near the aboral end, and this trait is retained into adulthood. The tentacles can be retracted into protective sheaths to keep them safe from the prying teeth of predatory fish. Along these 2 main tentacles, smaller finger-like structures called tentilla protrude laterally. These are used in hunting, but I'll talk about that more later on as well. The main point here is that the ctenopids are generally spherical, with rows of cilia running down them like the ridges of a pumpkin. Some species will be flattened in the same plane as the tentacles, with a body shape more like a puck or a disc. Some notable ctenopids include the Pleurobrachia pileus, or the sea gooseberry, which is a tiny little globule of transparent goo no more than an inch in diameter, but with tentacles that are almost 2 feet long. These are known to live in the cold waters of the North Sea and the waters around Europe, where they spend the night in shallower water, and then in the day they descend to go deeper into the water as they chase after their main prey item of crabs and barnacle larvae, and smaller species of copepods and amphipods. Another notable cetipid group is the Euplocamus genus, which are mostly found in the water column in areas with warm currents carrying heat from the equator towards the poles. These Euplocamus guys are wild, because among all of the ctenophores, they have one of the most developed nervous systems, including full axons that allow them to move and react quickly to stimuli. They have some of the most complex networks of channels carrying nutrients from the gut to the rest of the body. They have some of the largest cilia ever, not just among other ctenophores, but among all other animals. And they have mesoderm, a third germ layer in the embryo that technically makes them triploblasts. It should be pointed out that the ctenopids are paraphyletic. This is to say, other equivalently ranked clades originated with cetipid ancestors that underwent a major divergence. This is supported by the fact that at least 3 other major orders are both evolutionarily younger than the cetipids and possess a larval stage that has been described as cetipid-like. To summarize, this group was likely ancestral to at least 3 other groups, which retained a similar larval stage but developed different adult morphologies and lifestyles. They're different enough that we categorize them as a taxonomically equivalent rank, but just something different, so analogous to reptiles and birds, I suppose. Of these 3 divergent groups that came from within the cetipids are the lobata, for example. The lobata are moderately large, free-swimming ctenophores, whose bodies are characterized by 2 big lobes. These lobes are an extension of the body itself, developing to form a big cup around the mouth. An analogous structure in a human face would be like your chin, your cheeks, and your brow ridge growing forward to make a protective enclosure around your face and your mouth. It's sort of like that. Curiously, the lobata's tentacles do not trail behind them, but instead they lie coiled up along the inner surface of the lobes. With external cilia propelling the lobate forward, This big, two-lobed cup structure can scoop up all of the zooplankton in its way. The zooplankton get entangled in the tentacles held inside the lobes, and they're easily fed into the mouth. In this way, the lobata are somewhat passive, ponderous creatures, moving along like a biological, slow-motion aquatic scramjet. This is somewhat similar to the baroids. This second group of ctenophores within Psittacipita have larger bodies with a large opening at the front. In the lobates, the large opening is created by the lobes growing around the mouth. The mouth itself isn't particularly big. But in the baroids, this large opening is literally just a really big mouth. They don't have tentacles to catch prey, but instead they simply chase after their prey and consume them with a big, giant gulp. The baroids have a thin layer of sticky epithelial cells along their lips, and this helps them keep their mouth closed while chasing after prey, in order to maintain a hydrodynamic advantage. When the prey is finally within range, the mouth is opened, and groups of large, thick cilia inside the lips act like prehensile teeth to scrape and rake at the body of larger prey animals. For example, these heavy-duty cilia may be used to tear apart the soft, gelatinous bodies of smaller, unlucky ctenophores who happen to have been eaten by the baroid. The third of these major divergent groups is the Platytonida, and this is a more distinct group. The Platytonida really leaned into the flattened body form as they adapted to a benthic, seafloor-dwelling lifestyle. This evolutionary path really took them away from the other ctenophores. They stopped swimming as much, so almost every species lost its comb rows of cilia. They spend time on the ground, so they turn their throat inside out and use it as a muscular foot, akin to the foot of a slug or a snail, to slowly move around and slime their way over rocks and corals and sand. However, they retained the pair of tentacles, which can be seen streaming in the water current above their benthic form. Another weird body form can be seen in a much smaller offshoot branch, the Cestidae, which includes two species of deep-water ctenophores that resemble eels or snakes, sort of. Their bodies are massively reduced, having evolved into translucent ribbons with iridescent or bioluminescent edges. It's hard to tell just by looking at them where the mouth end is, and honestly, they look more like bits of plastic instead of actual living animals. Their tentacles are also reduced, being little more than nubs, and half of their comb rows of cilia are reduced and almost non-existent. Instead of exclusively using cilia to move, they use a combination of cilia propulsion and snake-like undulations. Okay, let's pause for a second to regroup and return to the topic of ctenophore evolution. I think a strong argument can be made for the case that ctenophores came after the sponges, by comparing the ctenophores to the cnidarians, or the jellyfish and their relatives. Molecular and fossil evidence is pretty clear that the jellyfish are the closest group to Bilateria, which is basically the rest of the animal kingdom, from clams to spiders to frogs to humans. Comparing ctenophores to cnidarians reveals the many similarities that these groups share, and thus supports the idea that ctenophores came after the sponges, and they are perhaps more closely related to the rest of the animal kingdom. Let's start with the traits that are shared between sponges, ctenophores, and cnidarians, so traits that they all have. Most obviously, all of them share not having a true digestive system. They don't have true digestive organs, like an esophagus, a gizzard, a stomach, a rumen, an intestine, or anything like that. The ctenophores, being translucent, fragile, bag-like creatures, have a vaguely defined mouth that in some species leads to a largely undifferentiated inner chamber where food is broken down. But this cavity isn't sealed off with a sphincter or enclosed in some way. It's quite open to the environment, at least compared to the guts of, say, a vertebrate. The bodies of sponges, ctenophores, and cnidarians are all derived from 2 germ layers in the embryonic state, whereas bilaterians are triploblasts, having 3 germ layers in the embryo. These germ layers go on to differentiate into all of the tissues of the body, and the lack of a 3rd germ layer is a big reason why these diploblasts are generally simpler organisms. In fact, there's even an ongoing debate right now about whether or not the ctenophores are in fact very basal triploblasts, based on the way their muscle cells develop, and the presence of a literal mesoderm layer in the Euplocomus genus. Research on embryonic ctenophores, however, is very difficult to conduct. It's a real challenge. So what this triploblasty might mean for their evolution is a mystery. Additionally, in each of these groups, the organisms have a layer of epidermal tissue that envelops the body and encloses a non-living, collagen-rich gelatinous substance. It's called mesohyl in sponges and mesoglia in the ctenophores and cnidarians, just in case you thought this wasn't confusing enough. This is where things start to get more and more different. It's where these groups start to show some contrast, and where a distinct split becomes apparent between the sponges on one hand and the ctenophores and cnidarians on the other. For example, in the sponges, the mesohyl is like a play and work area for the rest of the amoeba-like cells in the body to move through and do stuff in. The mesohyl is heavily populated with free-moving cells. But in the ctenophores and the cnidarians, the cells are much more grounded, literally. They've evolved more developed tissues, including basement membranes that other cells grow from, and stronger physical connections between cells that provide more cohesion and disallow this kind of free-moving amoeba-like movement style that you see so often in the sponge's internal cells. Ctenophores and cnidarians have tissues that are stiffened and made to be more durable, and their entire body becomes hardier and better able to endure the physical stresses of life in the ocean. The ctenophores and cnidarians have relatively few cells in the mesoglia, as their cells are by and large all bound up in actual tissues. The free-floating dynamic nature of the cells that we see in sponges, it's so reminiscent of pre-multicellular colony-like behavior. That trait was not preserved in the ctenophores and cnidarians. Further showing their divergence from the sponges, both the ctenophores and cnidarians are free-swimming creatures that are not anchored to the ground in one spot for most of their life. They swim around, they seek out and hunt down prey, and they evade predators of their own to the best of their abilities. All of this movement involves muscles, a specialized tissue type that enables an animal to move its body in controlled and coordinated ways. Both ctenophores and cnidarians have rudimentary muscles, which are totally absent in the sponge, along with almost all other types of tissue. The ctenophore muscular system is made up of a thin sheet of muscle fibers, positioned between the basement membrane and the rest of the cells in the epithelial tissue. A core feature of the ctenophores that distinguishes them from sponges and aligns them with the jellyfish and the bilaterians is the gastrial system. The sponges operate purely by diffusing nutrients across a layer of tissue, but more complex animals, including the ctenophores, have an internal gut or a cavity specifically for the digestion of food. The ctenophore gut is as simple as it gets, being little more than a cavity with a single opening. Compare this to the digestive system of a typical mammal, with its distinct and specialized segments, multiple compartments and glands, and the anus, which facilitates one-way transport of food. In ctenophores and cnidarians, and other organisms with a simpler two-way digestive system, food must be ingested and then excreted in sequence. This has fitness consequences, including an inability to eat more food until the current load has been digested and the waste expelled. Perhaps the most profound similarity between the ctenophore and the cnidarians that separates them from the sponges is the development of a primitive nervous system, with familiar nerve cells arranged in a decentralized nerve net that innervates the gelatinous body. Some of these nerve cells are distinct and communicate with each other through synapses, but other nerve cells are fused. They form a syncytium, which is like a large network of continuous cytoplasm with multiple embedded nuclei. Think of it like a huge cluster of connected neurons, all fusing their membranes and becoming one giant reticulated spiderweb-like cell with thousands of nuclei. This neural net innervates the entire body, but it's concentrated around the active structures, like the glands, the rows of cilia, the mouth and the pharynx, and the tentacles, if the species has them. Interestingly, there's also a concentration of neurons at the aboral end. This cluster of neurons seems to be independently derived from similar neural concentrations in the rear or the genital area of other animals, so it's not homologous, and they also have a more disorganized set of neurons in the mesoglia. These contain sensory organs that measure balance, using a grain of calcium carbonate suspended by the stiff cilia of these primitive pseudoproprioceptory sensory cells. These sensory organs are concentrated in the aboral cluster at the end of the body, this aboral ganglia. This is perhaps to prevent them from being soiled by the waste excreted from the mouth. One sensory function that the ctenophores are suspected of having is some kind of visual system. Perhaps nothing that can form images, but the simple detection of light, like the ability to tell the direction of sunlight and the time of day, or to detect the pattern and the color of biofluorescent lights on a potential mate. Based on genetic evidence in the species Nemiopsis leidii, the genes for the calcium-activated photoproteins that produce the bioluminescent effect are expressed at the same time as genes used for opsin proteins. Opsins are light-sensitive proteins. They're the same kind of proteins used in the eyes of virtually every other animal that has eyes. The fact that opsin proteins are expressed at the same time as photoproteins is curious and leads to some curious ideas about why that might be the case. The first idea that pops to mind is perhaps the bioluminescent photoproteins are being expressed as a form of flashlight, and to conserve energy, the eyes, so to speak, or the light sensitivity of the ctenophore, its ability to express opsins and detect light, it only expresses this when it also expresses the photoproteins, so it can see anything reflected in this brief flash of the bioluminescent flashlight. So it makes the flash, and it opens its eyes, metaphorically speaking, to see if anything's there. And then the bioluminescent effect fades, and the light goes out, and it closes its eye. It stops expressing the opsins, and its ability to respond to light stimulus is reduced, or it goes away for the time being. Now, all sensory organs are made of or built around nervous tissue, and the ctenophore neural system is relatively primitive compared to other more derived animals. It lacks genes for, and thus does not express, the receptors for a variety of neurotransmitters, including serotonin and dopamine. Instead, their nervous system uses a small selection of neurotransmitters, and it's really overdependent on glutamate. They have an unusually wide variety of genes for the proteins that synthesize, transport, and bind glutamate, and it seems to dominate the signaling activity across their neural system. Despite this variety of glutamate-related genes and the seeming over-reliance on glutamate as a neurotransmitter, the overall number of genes involved in the nervous system is low, lower than any other animal with a nervous system, which basically means every other animal other than sponges, which just don't have nerves. It's been suggested that the ctenophores possess a kind of bare minimum nervous system, with a small library of genes building a skeleton crew of very simple neurons. This basic neural system utilizes as few types of neurotransmitters as possible, like the letters in the Morse code alphabet, in order to send signals that sustain body processes and generate the most basic of behaviors, like twitching cilia in rhythm so as to move in a desired direction. There's two hypotheses that try to explain this. One idea is relatively straightforward, arguing that the ctenophores, being primitive basal animals, also have primitive basal neural systems. We shouldn't be surprised at this low level of complexity, like the over-reliance on glutamate instead of a wider variety of neurotransmitters, because these are basal systems that simply never needed, and thus never evolved, more complexity. The other idea is that the ctenophores had a more complex neural system, including a wider diversity of neurotransmitters, but this complexity was lost over evolutionary time. This ties in with fossil evidence suggesting ancient ctenophores had organs and more sophisticated tissues, which were then lost at some point in the lineage that produced the modern ctenophores. But none of this is conclusive, none of it is known for certain. It's currently unknown which of these hypotheses is true, if either of them are. Although, from what I can tell, the lost complexity hypothesis seems to be gaining ground. I could be wrong on that though, so don't quote me, take me with a grain of salt there. This is really an active area of research, so new data may come in and change our understanding. There's another type of cell that's actually unique to the ctenophores. You don't see them in sponges, you don't see them in cnidarians, and you don't see them in any bilaterian animal. These cells are called coloblasts, and they're specialized for hunting prey in the same way that hanging flypaper hunts bugs flying around your house. The coloblasts are mushroom-shaped cells, with the upper cap portion being coated in small, bubble-like granules. These granules break apart on contact with a prey animal, and they release a gooey mixture of sticky adhesive chemicals. A prey animal that gets tagged by coloblasts will quickly find itself entangled in the tentilla and the tentacles of the ctenophore. The prey animal will exhaust itself trying to escape, and eventually become helpless foodstuff to be ingested and dissolved by the ctenophore predator. So, for a typical ctenophore, a day in the life involves a lot of swimming. Not with fins, but with cilia, usually 8 rows of pigmented ciliated cells arranged in visible stripes along the outer surface that beat in unison to propel the translucent creature through the water. These rows of cilia have the appearance of combs, for which the comb jellies are named. Most cilia that you'd see in any other cell, like those in your digestive tract, are small, but the ctenophore's locomotive cilia are freakishly long. These usually beat away from the mouth, so the ctenophore is pushing forward, mouth first, to scoop up food particles as it moves along. However, the cilia can also beat towards the mouth, to move the body backward, which the ctenophore would do when trying to maneuver in tight spaces, when it's trying to adjust its attack vector when approaching prey, or when it's trying to dodge and evade a predator. The ctenophore, swimming mouth forward, envelops its prey with or without the use of sticky tentacles, and it traps it in the pharynx, or the upper throat area, where it's broken apart and dissolved by a slew of digestive juices and muscular contractions that are analogous to peristalsis. This dissolved mass is a slurry of nutritious gore, which gets washed into the channels by the action of the internal cilia lining the gut, and then it gets absorbed via diffusion by the epithelial tissue. Unfortunately, these digestive enzymes remain understudied. We don't know what kinds of enzymes are involved and what genes produce them. This information is valuable because it could provide insight on their evolutionary relationship with other animals. Like if they share genes for digestive enzymes with cnidarians that they don't share with sponges, well, that's more evidence in support of the idea that ctenophores emerged after the sponges, and they're more closely related to the cnidarians and the rest of the animals. But if their digestive enzymes are more similar to the sponges, like say every enzyme is an obvious homologue with something in a sponge and not something in a cnidarian or a bilaterian, well, that would complicate things, and it would give us reason to pause and reassess, or at least refine the sponge-first idea. But enough about these comparisons with other early metazoans. Let's just focus on the ctenophores from here on out, as we cover their ecology and their life cycle, and what's unique to them alone. We've already talked a lot about their evolution, and their relationship to other animals, their diversity, or lack thereof, their anatomy, from their body form to their cells and tissues. We've talked about their movement and hunting styles, their neural system, and their digestive system. This description included scattered references to a diet heavy on zooplankton, like tiny copepods, amphipods, krill, crustacean larvae, mollusk larvae, fish larvae, and so on. But they'll also eat smaller ctenophores and even cnidarians. There's even a ctenophore genus, Heckelia, whose species prey on jellyfish, and they salvage the jellyfish's nematocysts, the venomous cells used for stinging, to integrate them into their own tentacles. These tiny creatures that get vacuumed up by roaming ctenophores, like the copepods or the krill, are themselves predators of phytoplankton and single-celled algae. These microbial primary producers are abundant, and their metabolic activity, like fixing carbon and making oxygen, is critical for chemical cycling and the health of entire ecosystems and all of the life within them. The copepods feast on phytoplankton, causing their own populations to surge, while the primary producers suffer the microbial version of extirpation. This is bad for chemical cycling and ecosystem health, as it can lead to anoxia in bodies of water and hits to food web stability. By filter feeding on copepods and the like, the ctenophores keep these small crustacean populations in check, and secondarily protect the phytoplankton and the general stability of the food web and the ecosystem at large. But we must keep in mind, they also eat fish larvae too, and there have been at least 3 documented cases in Europe of ctenophores colonizing a new area, which all subsequently saw declines in its local fish populations. The ctenophores created a higher rate of attrition among fish larvae. Fewer fish became adults who reproduced, leading to smaller and smaller generations, and a decline in total numbers. Now the ctenophores, in turn, are preyed upon by a surprising number of animals. For example, in those 3 cases where ctenophores colonized new areas and disrupted the fish populations, they were followed by another ctenophore that preyed on the smaller ctenophores. This second species put a downward pressure on the first species, reducing its population size and its impact on the fish and the local ecology, kind of like a marine version of reintroducing wolves to Yellowstone to handle all of the deer eating all the vegetation. On this note, baroid ctenophores, those with the huge mouths, typically prey on smaller, slower ctenophores. It was originally thought that ctenophores weren't really part of the food chain, as they were seen as largely empty, nutrient-poor, watery bags of goo that made for an unsatisfying meal. Presumably, they tasted like nothing, and they delivered almost no calories. It was a waste of energy to even turn your head to bite at them or chew them up. This mistaken view was backed up by the purported lack of evidence of ctenophores in the guts of predatory animals, but this is easily explained as the ctenophores being pretty soft and mushy and extremely quick to break down and become indistinguishable from any of 1,000 normal secretions or slimes or goos or whatever else in the predator's stomach. It has since been observed and understood that ctenophores are perhaps not an absolutely vital part of the food web, but an option of counterintuitive importance for animals like crabs, fish, and jellyfish. Ctenophores are slower and easier to catch than similarly sized prey, like tiny fish and shrimp and crab, and they can be digested much more rapidly and with less energy investment to get the whole digestion process started. A fish or a sea turtle gobbling up a few dozen ctenophores is going to be satiated for the time being, and well-nourished for at least a few hours. Even herbivorous fish have been observed slurping up ctenophores, like they're some kind of tofu for fish. Jellyfish are particularly destructive enemies of the ctenophores. Both groups can develop large populations, like clouds of millions of semi-transparent tentacled bags of slime. Enormous populations of jellyfish can descend upon the ctenophores in an area and wipe them out for some period of time until the survivors regenerate sizable populations again, or new ctenophores move in and try to survive on their own. This brings us to the final topic of today's episode, the ctenophore life cycle. In this regard, the ctenophores are super interesting animals. They're really wild. The ancestral ctenophore was a hermaphrodite, which means that a single individual can produce both eggs and sperm from specialized gonad structures mounted under the comb rows. This is a good spot for them, as the comb rows are highly metabolically active and they're prioritized for nutrient delivery. Gonads being positioned near the comb rows means that all of these gametogenic cells are well fed and able to build new cells and new genomes with an ample pool of chemical resources. This already distinguishes them from the sponges, who produce eggs through totipotent cells and sperm through mutated choanocytes. The ctenophore hermaphrodite trait has been passed on to all of the ctenophore descendants, albeit with some important modifications. For example, some species are true simultaneous hermaphrodites. They possess both ovary and testy analogs, and they can produce both eggs and sperm at the same time, or in the same life stage. Other species of ctenophore are sequential hermaphrodites, which means they produce one type of gamete in one part of their life, and then at a certain time or stage, they cease to produce that gamete and they transition to producing the other type. These hermaphrodite species are capable of asexual reproduction through self-fertilization. Surprisingly, there are 3 species, all of them lobates, that are known to have evolved distinct sexes who only possess a single type of gonad during their entire lifetime. 2 of them belonging to the Iceropsis genus, and the third belonging to the Bathysauro genus. In any case, the sperm and the eggs are released through pores in the epidermis, where they flow into the external environment. This external fertilization is inefficient and messy, and thus it requires a huge amount of gametes. Think something like tens of thousands of eggs and several orders of magnitude more sperm. Much like the fish, the scale of this external fertilization can potentially lead to hundreds or thousands of fertilized eggs. And when you're talking about a mating population of hundreds or thousands, you can see how a new generation of ctenophores in any given area may start out with millions of offspring, who are then heavily preyed upon by all manner of aquatic creatures and attrited by environmental and physical forces. And yet, in the next generation, there can be millions more offspring. The platytenids show strange exceptions to these rules. These are the flat-bodied, ground-based ctenophores that are pretending to be some sort of starfish or sea slug. I describe them as like a disc or a puck. These platytenids can reproduce sexually like other ctenophores, but they can also reproduce asexually through a strange type of budding process that other ctenophores cannot do. Their juveniles are free-swimming like other ctenophores, but then they settle down in adulthood for this flat, ground-based lifestyle. In other species, the juvenile grows directly into the adult form. There isn't a dramatic metamorphosis event where the entire body is radically restructured in a short period of time, like you'd see in a crab larva turning into an adult crab, or a caterpillar turning into a butterfly. The ctenophores don't have this. They just grow bigger from juvenile throughout adulthood. This is where we see a particularly strange property of ctenophore reproduction. A large number of species, such as many lobates and cynipids, go through a really weird, interrupted, 2-stage reproductive growth process. As juveniles, the ctenophores can produce and release a small number of gametes. As they mature, they enter a period where they seem to be mostly focused on their own physical growth, and they stop producing gametes. And then, when they become adults, they start producing and releasing gametes again. This may be an evolved strategy for surviving challenging environments, as it encourages rapid sexual replication and genetic admixing. There's even a response that's been seen in adult ctenophores where challenging environments, like a lack of food, will lead them to stop producing gametes and reduce their body size until food is plentiful again. This interesting kind of pseudo-dormancy lets them endure periods of starvation and stress and recover quickly when resources become available again. Well, I think I've touched on every major topic of ctenophore biology. This was a surprisingly long episode for such a small clade, but they're just so weird and interesting that I couldn't help but take a closer look. If you enjoyed learning about ctenophores, hit that like button, or give the podcast a 5-star review. If you want to listen to my upcoming episodes on the Cnidarians and all the other animals, then subscribe to the channel. This stuff really helps me in the algorithm, and it makes it so more people can find the show. If you want to support the podcast in other ways, check out the official store, or become a Patreon member at a tier of your choosing. I always appreciate anything you guys do to support the show, so please keep at it. Tell your friends, tell your teachers, share the word. And as always, thanks for listening.